You've probably heard a million times that DNA is the blueprint for life. But what if I told you that blueprint isn't the whole story? What if there's a hidden layer of control, a whole other system, that tells our genes when to speak and when to stay silent? Today, we're diving in and unlocking that code.
I mean, really think about it. What if there's another layer of information sitting right on top of our genes? A dynamic, flexible layer that controls which parts of the blueprint actually get read and when. This hidden world is where some of the most mind-blowing biological discoveries are happening right now.
And this field of study has a name you're going to want to remember: epigenomics. It's all about these chemical tags and structural changes that, and this is the key part, don't actually alter the A's, T's, C's, and G's of our DNA, but they have a massive impact on how our cells function, basically acting as the switches that turn genes on or off.
You know, a really powerful way to picture this is with a river. Think of epigenomics as the upstream regulator. It's the source of the river. Everything that happens downstream, all the processes in our cells, is governed by what's happening way up at the source. It is the ultimate biological control panel.
But here's the thing, this control panel isn't just one big on-off switch. No, it's a whole series of them, all working together. We're talking about things like DNA methylation, which acts like a dimmer switch for genes. Then there are histone modifications, changes to the proteins that our DNA is wrapped around. There's also chromatin accessibility, which is all about how tightly or loosely that DNA is packed. And it doesn't even stop there. We've got non-coding RNAs and even modifications on RNA molecules themselves. Every single layer adds another dimension to the story.
So, if all these layers are working in concert, just looking at one of them in isolation is kind of like trying to understand a symphony by only listening to the violins. You miss the whole point. So that's the big challenge for researchers, right? How on earth do you see everything at once to get the full picture?
Well, the answer is this incredible strategy called multi-omics integration. It's all about combining and analyzing data from all those different layers, the genome, the epigenome, the transcriptome, you name it, to finally see how they all connect and influence each other.
And scientists have two main strategies in their playbook for this. First, you've got direct correlation analysis. Think of this like finding matching puzzle pieces. You just overlay two different data sets and look for where they overlap to find some really promising targets. Then there's the indirect validation method. This one is more like following a trail of dominoes. You're trying to map out the entire cause and effect chain of events, from one change all the way downstream to its final impact.
Okay, that sounds great in theory, but what does it look like in action? Let's dive into some actual research and see how scientists are connecting these omics dots.
All right, for our first case study, we're tackling a huge question in cancer research. How do some cancer cells not just survive, but actually thrive in the super harsh, low-oxygen environments inside a tumor? I mean, this is a critical survival mechanism that researchers are desperate to understand.
This is so cool. By putting RNA modification data and protein data side by side, they found a key player, a protein called YTDF1. They figured out that when oxygen is low, this protein ramps up a cellular recycling process called autophagy. Basically, it allows the cancer cells to eat parts of themselves to survive. It's a perfect example of connecting an epigenetic mark directly to a critical survival function.
All right, case study number 2. This one is about something that sounds like it's straight out of science fiction: cell reprogramming. We're talking about the almost magical process of turning one type of cell, say a skin cell, into a completely different type, like a stem cell. The big question was, how could a single protein possibly kick off such a massive transformation?
Well, to figure this out, they had to pull out all the stops, a massive multi-omics effort combining 4 different data sets. And they uncovered this amazing cascade of events. It turns out a protein called GLIS1 first changes the cell's metabolism. That metabolic shift then creates the fuel for epigenetic changes to the histones, which then opens up the DNA and activates the genes needed for that cell to become pluripotent. It's this incredible epigenome, metabolome, epigenome story. Just amazing.
Okay, those stories are just incredible, right? They really show the power of seeing the whole system. But it brings up a really practical question. If you're a scientist working in a lab, how do you actually do this? How do you get access to all these different layers of information?
The way they do it is by using a whole suite of powerful epigenomic sequencing services. These are the highly advanced instruments and methods that let researchers read those epigenetic marks across the entire genome and transcriptome with unbelievable precision.
And this is basically the researcher's toolbox for epigenomics.
On the DNA side, for instance, you've got a technique like ATAC-seq, which can map out exactly which parts of the genome are physically open for business and ready to be read. And then on the RNA side, a method like Upstream can pinpoint those exact chemical tags on RNA molecules that we just saw were so important in that cancer study. For scientists to pull off this kind of complex multi-omics research, they rely on specialized services that offer this full suite of tools, and that's exactly what specialists like Genomics provide.
So let's pull back for a second and just talk about the big picture here. Why does all of this matter so much? What does this technology really unlock for us?
Well, it really comes down to 4 huge advantages. First, you get these comprehensive insights, a full map of the entire epigenetic landscape. Second, the sensitivity is just incredible. We can detect even the tiniest, most subtle changes. Third, it's high throughput, which means we can analyze thousands of genomic regions all at once. And finally, you get this amazing dynamic resolution to see how these marks change over time or in different cells. It is a complete upgrade in our ability to see biology in action.
So when you put it all together, when you integrate these multi-omics approaches with these powerful sequencing tools, you completely change how you see the genome. We're moving away from seeing it as some flat, static instruction book and toward understanding it for what it really is, a dynamic, responsive, multi-layered system, a living code that adapts and changes.
And that just leaves us with this one final, really exciting question. We are really just scratching the surface of this new layer of biology. So as these tools get even better and more integrated, what fundamental secrets about life are we going to unlock next? The possibilities, they're just extraordinary.